はい、演者は、メリット、サコーヴィッツ先生です。メリメリットさんはですね、1990年にハーバードメディカルスクールを卒業して、そこで MGH、マサチューセッツジェネラルホスピタルでレジデントをやって、そのまま、96年から同じ場所でファカルティをされています。そのまま、2012年からは、プレジデント、でチェアマンも行っているということで、この領域の、ALS の治験の、アメリカにおけるリーダー、第一人者に、なっているかと思います。個人的には私と森田さんが、MGH 留学してた時に、ちょうど一緒に研究していた仲間でありまして、もうずっと昔の話ですけど、大変懐かしい、思いがいたします。それでは今日はビデオになると聞きました。よろしくお願いします。
Hi, I'm Merit Sokovich. I'm the chair of neurology and director of the Healy and AMG Center for ALS at Mass General Hospital and the Julianne Doran Professor of Neurology at Harvard Medical School. I'm really thrilled to be here to give an update on ALS therapy development, where we are on new treatments and updates on clinical trials. I have a slide on my disclosures that are listed here. So ALS is considered a rare disease, but in the United States, at any one time, there's about 25 to 30,000 adults living with it, more than 500,000 people globally. The lifetime risk is actually pretty high. It's 1 in 350 males over their lifespan will die from ALS, and one in 440 women. Average onset in 55 years, but this can happen in people in their 20s as well as people in their 90s. 10 to 20% of people have an identified genetic mutation. For the rest of the people, while we call it sporadic, there's thought really to be about a 50% heritable factor to it of risk factor genes, and a lot of those are being discovered. So we know a lot more about the ALS biology, and this is helping us develop better treatments. So a lot of the genes that cause the familial form cause defects in autophagy, in RNA metabolism, in cytoskeletal and axonal transport, in mitochondrial function, in inflammation. And this tells us that these are targets for people with ALS to develop treatments. There's been a lot of therapy development to target these different pathways. For example, there have been drugs looking at excitotoxicity, and we have one marketed drug, riluzole, that slows progression by 10%, that acts on excitotoxicity. And there's many more drugs in development that target hyperexcitability, particularly drugs that work on potassium channels. There's great interest in looking at the oligodendroglia and their role in contributing to cell death in ALS. There's a lot of interest also in the nuclear pore biology as perhaps some of the earliest changes in the motor neuron and a really important target. And there's development with Ionis Pharmaceuticals and Hopkins to work on an antisense oligo that targets a nuclear pore protein called CHIP7. Many companies and academics are interested in axonal transfer and degeneration. There's a few companies developing treatments to try to inhibit the loss of axon integrity or to accelerate axon repair. Also approaches mitochondria and metabolism, in particular Radicava, clean nanomedicine. Many neuroinflammation drugs have been tried and have not worked yet. This is very complex, and it might be that one needs to have different and combinatorial approaches to tackling neuroinflammation. Now, the Northeast ALS Consortium was formed in 1995. This is a group of now over 150 centers that are global, and we'd be thrilled to have many more sites from Asia as part of our consortium. This is a group of scientists, clinicians, clinical trialists, everybody who touches people with ALS working together to try to solve ALS. We do many trials together. We've had over almost 80 studies together, 50 interventional trials. We've launched the first ALS platform trial together. We share all the data that we collect from clinical trials in an open source database called PROACT. We have many, many samples from participants in those trials that we share globally. We have a single ethics review so that we can speed up study startup, and we do a lot of training of young students and faculty, investigators on outcome measures, how do you lead a trial. We also train people living with the illness and their spouses to be research ambassadors. We have a science advisory board that meets with companies, meets with academics to give advice on preclinical models and trial design. We've had successful transitions in leadership. Dr. Scheffner and myself were the co-founders and initial co-directors, and now it's led by Dr. Jinsey Andrews and Dr. James Berry. Now we've learned a lot about ALS trials, and we are really poised to have huge success. We have outcome measures, we have trained sites, we know how to do trials. And as we understand more about the biology, we're getting better and better drugs. So since 1995, we've had riluzole as a treatment, and that has reproducibly in every trial that has looked at it, slowed the disease progression by about 10%. Initial studies were large though, over 1,000 people and looking at survival. The Darivone came in 2017 in the US, earlier in Asia, and that was approved on a smaller study looking at function and taking people early in the illness with fast progression as a way to enrich the population to be able to tell an effect in 6 months. And that was a really transformational study that I think has changed how we do trials. After that, sodium phenylbutyrate and tirsodiol by Amylyx was combined also in a study that looked very much like the Darivone study, and that was positive in the United States and brought to market on that one study. Unfortunately, the phase 3 study in Europe was negative, and there are many reasons for that. One of them could be that the drug really doesn't work. The other one could be that the design really changed a lot in that second study, but that is now no longer on the market. And most recently, the first targeted gene therapy trial for one of the genetic forms of ALS caused by mutations in SOD1 was approved based on a change in a biomarker. So the first accelerated approval on a biomarker for people with ALS. And this is really an amazing advance for ALS and something I'll tell you more about. So some of the themes in trials today are that instead of taking very broad populations and doing long, large studies, we're taking much more homogeneous, smaller group of participants and really testing drugs in shorter time. And that's so important in such a rapidly fatal illness like ALS. We're focusing on biomarkers, the approval of a drug based on neurofilament, like tofersen, a huge step forward for ALS clinical trials. And at least in the United States and Canada, a single pivotal trial is sufficient for approval. Now, we always want to have confirmatory trials, but it's really the balance of when make a drug available for people versus doing confirmatory trial. So I want to spend a little time on the data for Qalsody or tofersen, which is the SOD1 trial drug, and then I'll talk about this new innovation on platform trials that's not only really relevant for ALS, but relevant for many neurological illnesses now, as we reach a time of real advances in neurotherapeutics. So the very first patient I ever took care of is pictured here, Susan. She carried the gene for SOD1. And at that time in 1994, there were really no treatments. I'm very excited that in 2023, in the United States, the first therapy, an antisense oligo, was approved for her form of the disease. Now, the gene was discovered in 1993 by Dr. Robert Brown and his colleagues. Mouse model followed soon after, and really we needed to wait for the revolution in how do you modify genes in the people in the brain and the spinal cord to get this treatment. The first antisense oligonucleotide study was actually done in SOD1 patients in 2011 and 12, but that particular version of the antisense had some toxicity in animal models, so a newer version was made, which is Qalsody. Now, this drug is on the market in many, many countries. In the trials, 40% participants had actually improvement in their strength. This is the first time in really the history of ALS that a drug has led to some people stopping progressing and getting better, and it tells us it's possible. And now we've seen that same phenomena in gene-targeted treatments for FUS ALS. We also saw a 50% lowering of a biomarker called neurofilament that led to the accelerated approval, and the lowering of that marker also predicted clinical response. So the design of the trial, the phase 3 trial, was relatively small. 72 people received active drug, 36 placebo for 6 months, followed by everybody getting active drug, and that open label extension is still ongoing. We saw good target engagement. The blue line is the placebo group, the green, the group that got the tofersen in the double-blind period first, so what we call the early start treatment. We see immediately, a significant lowering of the SOD1 and the protein in the CSF, and we know that this amount of lowering in the CSF is connected to 70 to 90% lowering in the spinal cord, and we know that from animal modeling. We also saw this dramatic lowering of plasma neurofilament and CSF neurofilament. We saw it about 50% by week 12, even more by week 28, and sustained during open label. We saw the people who started the drug 6 months later, the delayed start group, also having that lowering later. And also we had lots of analysis that showed that this also was related to clinical effect. This is the clinical data, ALSFRS, SVC, and handheld dynamometry, and you see in each of these figures that the curves start to split with the group on early treatment, doing better than those who got the treatment later, in particular in SVC and handheld dynamometry, and you start to see people plateauing, particularly on their strength, 8 to 10 months into the treatment. We looked at how many people got better, and this was defined by people who had either no change or a positive change in any of these measures. And you can see in particular that people who were in what we call the slow progressing group, so people who had low neurofilament, more likely to have this recovery, and in particular people also who started the drug earlier were more likely to have this benefit. So a lot of learnings from this trial. So just to summarize, tofersen administration led to about 60 to 70% reduction in neurofilament, indicating slowing of axonal injury and neurodegeneration. Earlier initiation of tofersen led to continued slowing of decline in function, strength, and quality of life. We saw improvement in function and strength in the subset participants, which is, again, just amazing to see. We saw a reduced risk of death equivalence, which I didn't show you here, but it's been written in the paper, and it was reasonably well tolerated. There were some serious neurological events, such as inflammation, transverse myelitis. They were rare, they were not treatment limiting, and they're managed with standard of care. So now I'm going to switch to the platform trial and why we decided to launch a platform trial and how we hope that this will eventually become a global platform trial as well. There's a huge pipeline of treatments in development for ALS. There's more than 300 companies. There's many trials going on. And whenever you have this many trials, it's much more efficient to have a platform trial approach rather than single study. So on the left, you see in blue are a couple drugs that have finished phase 2 trials and phase 3 trials that have positive results. Methylcobalamin, interleukin-2, prime C, CNN, AU8, and pridopidine. In red are phase 3 trials that had negative results. In the middle are active trials. But there's huge, what I call pipeline pressure. So the NEALS consortium got together and met with leaders in platform trials from oncology and put together the first Healy platform trial for ALS. And we really took a group effort to get this designed from our membership in NEALS many foundations, many companies interested in trying this approach. So what's the advantage of a platform trial? Well, it saves money, it saves time, and it greatly reduces the number of people on placebo. So it has huge advantages to traditional trials. It only makes sense if you have a big pipeline. So in a traditional trial, you test one drug at a time. It's kind of like building a football or soccer stadium, playing one game, and then taking the whole thing down. In a platform trial, you test multiple drugs and you keep adding drugs or taking drugs away until you find treatments. And you don't destroy or take down the infrastructure. You share the infrastructure. And that's where you get the efficiencies in time and money. And this is much better for patients living with such a serious illness. When we designed this, we got broad input. We went to the FDA to get their input. Very important that they understand it and they are supportive, which they were. We went to companies to understand what they might be worried about, what would get them excited about participating. We went to the patients to get their input on the design, went to the foundations and to our sites and our investigators. And we were very proud to launch this in 2019. We have a 5-star rating from patients that this is a very patient-centric trial and that they're excited about it. And we can see that by the very fast enrollment rates. Platform trials are really a win for all the stakeholders. So in a platform trial, you have a common protocol. So all the drugs in your platform trial, they do have to follow that same master protocol. There can be some flexibility around safety issues, but everybody has to have the same duration of treatment, the same inclusion criteria, the same outcome measures. Now you can add additional ones, but the basics all have to be the same. And so in 2020, we launched with the first 3 drugs. We call the drugs regimens. And each regimen has active drug and placebo, and we designed it to be 3-to-1 ratio. So 75% of people in each regimen get active drug and 25% placebo. Then we added another one, regimen D, and this is where you get the efficiencies. Because it's one protocol, to add a drug, you just amend the protocol. The FDA has asked for 30 days. The IRB, which is a central IRB, just reviews the safety of that drug. So it took, for example, 9 days to get IRB approval for regimen D, 30 days to get FDA approval, and then all the sites are ready. So it's very efficient, as opposed to a standard standalone drug where it might take 6 to 9 to sometimes 12 months to get a new drug trial off the ground. Then we added regimen E in 2022 and then F and G in 2023, and we're now working with 3 companies, possibly a 4th for 2025 to add several more drugs to the regimen. So 7 regimens so far, 70 plus sites, all in the United States currently. More than 1,300 people have participated in this, and we're very excited. So for each regimen, people are screened and then they're randomized 3-to-1, and the placebo-controlled trial for the first 7 drugs was 24 weeks, followed by open label extension until we have the results. The primary outcome measure is change from baseline through 24 weeks in disease severity as measured by ALS functional range scale and survival, and we have a bunch of secondary outcome measures. We are, for future regimens starting in 2025, going to go to 9 months for follow-up. We also want to learn about the illness. We want to learn if the drugs work, but we also want to learn about the biology of the illness. So in every regimen, people contribute DNA for whole genome sequencing. Everybody gets neurofilament and other biomarkers specific to the drug. We're developing some digital apps as well. And in the next regimens, we're also adding PBMC collection for iPSC development. So we initially wanted to take drugs that target different pathways. So regimen A targeted a complement pathway, regimen B, microglial activation, regimen C, energy metabolism, and regimen D was a sigma receptor agonist. Regimen E attacked autophagy. And then for F and G, they both had similar targets of integrated stress response and TDP-43 biology. So if someone comes into the platform trial, they're first randomized to one of the open regimens at that time. So if you came in the beginning, you'd be randomized to A, B, C, or D. Then there's a second randomization to drug or placebo. That's 3-to-1. And so people don't get to pick which drug they're in. So you really have to make sure you have equipoise between your regimens. And then you're followed for the 6 months. And the benefit, again, is that when you're done, let's say, with regimen A, you're comparing the active arm in regimen A to the shared placebo from all the other concurrent regimens. And so that you have really a very strong power. And then after someone's individual 6-month period, they are followed in open label extension until we have the results, but it also gives information like we saw in tofersen around delayed start versus later start. So we have results of the first 5. 2 of them have had positive results on going to phase 3. 3 were clearly negative. The negative ones are regimen A, B, and E. So zilucoplan, verdiperstat, and trehalose. Regimen C and D did not hit their primary outcome measure, but they had positive results on some of the secondaries, and including neurofilament, and are moving forward to phase 3 testing. Regimen F and G are fully enrolled, and results expected later this year in early 2025. So platform trials are a new innovation in neurology, not so new in clinical trials. We're now seeing platform trials in Europe as well, and in many other neurological illnesses. So it's really important. They're faster, they're more efficient, they're better for patients, and they're really here in neurology. We've learned a lot from the first couple of years. We're adapting the platform for 2025, and we're very happy about how it's going, and it enrolls very well. We continue to learn about the illness, and we're sharing what we're learning with the community as well as other fields. So I want to end and just thank you for listening to me. Thank you for an amazing group of investigators at NEALS throughout really the world, working together to try to cure this very serious and very difficult illness. And I remain very confident that we're on really the cusp of finding meaningful treatments for people living with ALS. And thank you.
はい、大変ありがとうございました。アメリカにおける治験の実情と、アメリカらしい大規模に、どんどん治験を組み合わせてやっていこうというお話でした。今日は残念ながらこれビデオですので、質問にはお答え、ディスカッションできませんが、何か質問があれば答えられるかもしれませんので、後ほどお寄せいただければと思います。それではこのセッション終了したいと思います。